Hello everybody, and welcome to the University of Kansas Ecology and Evolutionary Biology REU of 2017. During this REU, we were tasked to create a video of the REU students and their projects. The instructions for this video said that students must only spend 15 seconds describing their research and then answer a follow-up question. Let's see how well the students followed the instructions this time.
So, the main focus of my research during this summer is analyze the niche of the species of the genus Lynx to see if the climate change have an effect on the distribution of the species. That's basically what my project is about. The lynx is a feline like a cat, a small wild cat. There are 4 species: the Spanish lynx, Canadian lynx, American lynx, and Eurasian lynx. Those are the only species.
I'm developing a model in C that simulates an asexual population undergoing clonal interference. Clonal interference is when 2 or more beneficial mutations that arise in the same background because they can't recombine interfere with one another. And what's important to notice is that the beneficial mutation rate increases, but the rate of adaptive evolution in that population actually decreases.
Jamestown Canyon virus is a mosquito-borne virus that's widely spread throughout North America. Our research is trying to create a mathematical model that will correctly show the transmission of the virus and how it is affected by breeding times and temperature variations. It's an SIR model, and that shows how transmission happens between different species based on whether they're susceptible, infected, or recovered from the virus.
Jamestown Canyon virus is a rare mosquito-borne virus that's widespread throughout North America. And our research focuses on developing a mathematical model that can accurately predict its transmission over time based on the breeding peaks of mosquitoes and also temperature variation. Well, it's important because Jamestown Canyon virus can cause some pretty severe symptoms like nausea, vomiting, and swelling in the brain. So the results of our model will help future public health care officials to implement better control measures.
We are studying plant soil feedbacks by inoculating 3 grass species with a soil nutrient gradient from a previous field experiment to determine if microbial communities will mediate plant response to the fertilizer. Microbial communities are microorganisms in soil such as free-living bacteria or fungi. Their interactions with plants can either be pathogenic or mutualistic depending on the type of organism it is, and in the Beaver Lab our focus is on arbuscular mycorrhizal fungi and the symbiotic relationship it has with plants.
Parallelism is when related species have independently evolved the same trait. I'm looking at a group of plants called penstemon to see whether genotypic parallelism is controlling the phenotypic parallelism we see. Penstemon is a group of around 280 flowering plants that has independently evolved the same hummingbird pollination syndrome of red flowers 10 or 12 times. And I'm looking at a plant in penstemon that has magenta flowers to see whether it fits into the pattern of parallelism that's been seen in the genus already.
Hi, this summer I studied the relationship between arbuscular mycorrhizal fungi and abiotic soil properties like gravimetric moisture content, soil aggregates, and soil bulk density. I did this over a gradient of land use histories meant to mimic 3 different stages of a prairie restoration. All right. Arbuscular mycorrhizal fungi are really important to study because they have a mutualistic relationship with 80% of all plant species, and they trade phosphorus, nitrogen, and sulfur in exchange for carbon. This promotes plant biodiversity by competition and allows plants that usually wouldn't be doing very well to do a lot better.
Hello, I'm studying how nitrogen fertilization and haying disturbance affects the diversity and species composition of a native prairie restoration. We have 32 subplots, and half of them are fertilized with nitrogen, phosphorus, and potassium, and then half of them are hayed once a year. And every summer we harvest a section of each plot, and we individually count out how many species are in each subplot, and it's pretty fun.
This summer I looked at soil composition in relation to the productivity of Lespedeza cuneata. So what I found was organic matter had a negative impact on the productivity of Lespedeza. And in terms of land management, further research I would like to be involved in would be looking at how shifting prescribed fires from the spring to the fall affects the amount of organic matter for the next season, as well as the productivity of Lespedeza for the next season.
Hi, for my project this summer, I'm looking at cryptic species of anole lizards to see whether populations that are genetically distinct are also morphologically distinct. So I'm counting scales and I'm looking at body size to see whether these different populations can be elevated to the status of species. Cryptic species are species that are, to the naked eye, identical, but actually have underlying genetic or morphological differences that make them distinct. By identifying cryptic species, we can further our understanding of the biodiversity on this planet and be better equipped to protect said biodiversity.
For my project this summer, I'm working to find out whether the reproductive activity of female Anolis cysticus is more affected by rainfall patterns than the reproductive activity of males of the species. Studying seasonality in Anolis contributes to our general knowledge of reproduction across species, and it also shows us the importance of climate in maintaining biodiversity.
My project involves looking at how piRNAs that are transmitted through the female germline of fruit flies affects gene expression in or across generations. piRNAs are small RNAs that target transposable elements and suppress their activity. However, sometimes genes get turned off in the crossfire, and that's what my project is focusing on.
Saprianus indianus is an invasive Drosophila species, and this summer I tested how wing ablation affects their courtship behavior and also how the order of mating affects the number of progeny that they produce. Wing ablation is the cutting off of their wings. So basically what I did was I cut the wings off of several flies and then compared them against control flies that had both of their wings.
Okay, well, my name is Maria and I am doing research in Dr. Smith's lab with spiders, and we are looking at the gravitational effects on web asymmetry. And web asymmetry is when the top of the spider web is smaller than the bottom half below the hub of the web.
Hello. My project this summer, I'm working on a complex species,
Right?
Jamestown, sorry.
Okay. All right. Okay.
What are you working on this summer?
I don't know.
What is your research over?
Why can't I say this? Okay.
Oh no, I forgot. Hold on.
Stay okay.
From a previous experience, oh my goodness.
Clonal interference is how when two or more
Niche analysis of the genus length. Okay, no.
Michaela, what was your project this summer?
My project this summer was making sex tapes for flies.